Hello students, I hope you're all doing fine. Welcome to today's session. In this session we are going to solve the solutions for Aiet 2 examinations for Neet paper. So you can check the test code, that is JSMT0224. So this should be your paper. So let's begin with the first question. Okay. So first we'll solve with question number 104. So the question says assertion. When incised, the incisions do not touch the midrib in a simple leaf, and the reason is the bud is present in the axil of the petiole in both simple and the compound leaves. And you have four options. Both assertion and reason are true, and the reason is the correct explanation for the assertion. Next, both assertion and reason are true, but the reason is not the correct explanation for assertion. Next one is the reason is true and the assertion is false. Both the assertion and reason are false. So let's look into this question part by part. The first one is the assertion part. So in the assertion part it's given when incised. So if you look at the leaf, okay, the leaf lamina is like this, correct, right? Okay, this is the leaf, and you have the leaf lamina. This is the leaf lamina. So in some of the leaves you can see that there are some ridges like this, okay, that goes across. Okay, I can draw a leaf like this, and you can see here the leaf has some ridges in the lamina like this, correct? No, you can see. So if these ridges do not touch, they are called as a simple leaf. If the ridges touch and then they break the leaf into small leaflets like this, for example, you can see the curry leaves or the neem leaves. Okay, if they break them into small leaflets, they are called as a compound leaf, correct, right? So here what you can see here, they have given that when incised, the incision do not touch the midrib in a simple leaf. That is absolutely true. So that is why our assertion is correct. So let's look at the reason. So the reason for this is absolutely not correct. But anyways, let's look at the statement. The bud is present in the axil of the petiole in both the simple as well as compound leaves. Yes, this is absolutely true. In both simple and compound leaves, you can see that there is a bud present at the axil. And what is this bud? This bud can develop into a leaf bud can develop into a leaf, or this bud can develop into a floral buds, or it can lead to the formation of flowers. So what is the right option? In this statement, both assertion and reason are true, but the reason is not the correct explanation for assertion. So that is why we go for option B. Okay, so let's go for the next question. Our next question is a very interesting question. So I think we solved this similar situation even when we are talking about principles of inheritance and variation. But anyways, let's look at the question. A couple has blood groups AB and A. What could be the possible phenotypes of their children? And the given options are, option A says A, B and AB are possible with the children, and the children will be having AB only in option B. C says O only, and the D says A and B only. So let's look at what's possible. So a couple has AB and A. So let's assume that the father has IA, IB. So these are the alleles of father, and he marries the mother with IA. So let's assume that she has the homozygous type, that is IA. So this is one possibility, and the other possibility is that you have the father, that is IA, IB, which you cannot change, and then you have the mother with IA and small I, correct? No, even then she will be having the A blood group only. So what would be the possible babies here? IA, correct? IA, comma, then you have IA, and then you have IB, IB, then you have IA, IB. So from here it's done. Let's go for the next one. So here the possibilities are IA. Next one you have IA, small I. Then you have IA, IB. Next you have is IA, IB, small I. So what do we get here, students? First, here we get A blood group. Here also we get A blood group. Here we get AB blood group. Here we get AB blood group, correct? And then here we get A blood group. here we get Ia small I is A blood group, then Ia IB is B blood group, and IB small I is B blood group. What do we get? We get A, we get AB as well as B blood group. So where do we have an option? Yes, option A is the correct one. That is A, B and AB is possible for the next generation to have these blood groups if the mother and the father is having AB and A blood group. Did you understand this? Okay, so let's go for the next question. So 107 question. Read the following statements. Cells of root meristematic zone have dense cytoplasm. Pineapple and a pineapple are the examples of sucker. Fleshy bud is called as a bulbil. Root cap is absent in hydrophytes. Which of the following statements are correct? All of these are correct. All except B. B and A are incorrect. Okay, and then B and C are incorrect. So let's look at each statement one by one and we'll analyze which is correct, which is wrong. So the cells of root meristematic zone have dense cytoplasm. This is absolutely true. Why? Because they are very actively dividing cells and they require energy. So that's why the cells are quite dense with respect to the meristematic zones. Meristematic zones are the zones of active cell division. You can see they have shoot meristematic tissue and you have the root meristematic tissue. So in order for them to divide fastly, they require the reserve food energy, which you can see as the counter for how the cytoplasmic thickness is. If they have this dense cytoplasm, that means these cells are actively dividing. Okay, next one, pineapple. Okay, are the examples of sucker. So you have pineapple and then you have jasmine. Okay, so pineapple and jasmine are the examples of sucker. So even this particular statement is true. Okay, next one is a fleshy bud is called as a bulbil. Yes, what is a fleshy bud? Usually a bud is something that acts as a storage organ. So they are stored. They store food as well as they perform reproduction, basically vegetative propagation. They participate actively in vegetative propagation. Okay, they are called as bulbils. So this is also true. A root cap is absent in hydrophytes. Yes, the root cap is absent in hydrophytes. So all the statements are correct. Okay, so that is why we go for option A. Okay, next one. Arrange the given enzymes in progressive correct sequential manner during DNA replications on template strand with polarity. Okay, so now what is happening? We have to see what are the sequential events that happens during the DNA replication. So let's see here. Generally what happens, we have a double-stranded DNA, correct? I will simplify this process. So when we have a double-stranded DNA, the first thing that has to happen is the site of replication is recognized, okay, by the helicase. The helicase comes and starts the replication at the ORI site, that is the origin of replication. So when helicase comes here, what it does is it unwinds the strands, okay, it unwinds the strands. Now you can see that based on how the strands run, like 5' to 3' direction or 3' to 5' direction, you can differentiate the strands into leading strand and lagging strand. Usually the strand that goes from 3' to 5' direction is called as the leading strand. Why? Because the strand that is being synthesized newly runs from 5' to 3' direction. So 5' to 3' direction synthesizes DNA strand in a continuous fashion. So that's why we call it as a leading strand. You understand? Okay. So now helicase came. Now after the unwinding of the DNA, now once the DNA is unwound, now the DNA becomes quite unstable. Okay, the DNA becomes what? Quite unstable. So when they become quite unstable, what happens here is, so you need to make sure that the DNA does not come back and and form the bonds again with the counter strand. So that's why these exposed hydrogen bonds are stabilized by SSBPs. So SSBPs are single strand binding proteins. Okay, they are called as single strand binding proteins. So after SSBPs come, so one rule that we have to understand here is, The DNA polymerase is an enzyme that actively participates in the synthesis of new DNA. But any DNA polymerase do not have the capacity, I insist here, they do not have the capacity. Do not means they can't. What can't? De novo. We call it as de novo. De novo can also be translated as cannot synthesize on their own. On their own. What does it mean? That means that the DNA polymerase requires some help. So what help we are talking about? To start the DNA replication, it requires a free 3' OH. Okay, let me write it here. What do they require? They require a free 3' OH. So a free 3' OH. So a free 3' OH means, you know that in your nucleotide structure, okay, if you take adenine, guanine, cytosine or thymine, you can see that at the 5th position of the carbon atom you have a phosphate group, and at the 3rd position you have a OH group. So you already need a particular nucleotide, okay, with a free OH in the 3' group. Okay, at the 3' you need a free OH. So this is called as free 3' OH. So who provides this free 3' OH? A free 3' OH is provided by DNA dependent RNA polymerase. So this DNA dependent RNA polymerase is an enzyme which we commonly refer it as primer. So this primer has the capacity to start the replication from first base pair. So it comes and it lays down a short RNA sequence, a very short RNA sequence. So this primer, what does it do? This primer comes to the 3' position of the leading strand, okay, and see, this is your strands that are split, and this is the 3' to 5'. The primer comes now and lays down a short primer sequence. Now what do we get here? Here we get a 5', here we get a 3' end. What is this primer? This is an RNA primer. Got it? No? This is an RNA primer. Now what happens? Now the DNA polymerase can come and start synthesizing the new daughter DNA in what direction? In 5' to 3' direction continuously. Okay, the situation is same in both leading and lagging strand, but there is a discontinuity in the lagging strand. Anyways, okay, so once we have this DNA polymerase, now what do we have? We have a situation with respect to the lagging strand. In lagging strand, what happens? The parental strand of the lagging strand goes from 5' to 3'. So as the helicase is unwinding in this direction, now what happens? A new strand is exposed every while, correct? No, a new strand gets exposed every while. Now the primer has to do an additional function. The additional function of the primer is to lay a short RNA primer. Why do they have to lay a short RNA primer? Because the more new DNA strands exposes, the more they lack the free 3' OH. You see here, I have written the short primers. You can give them the, this is 3', this is 5', this is 3', this is 5', this is 3', this is 5', like this, okay? A short RNA primers are laid. Next, these primers are again extended by a DNA polymerase, correct, right? A DNA polymerase will extend these primers. Once this is done, again these RNA sequences have to be removed, right? So these RNA sequences will be removed and again will be replaced by a continuous DNA. Now if you look at the DNA in the lagging strand, you see this is the parental strand. If you look at the newly synthesized DNA in the lagging strand, they are not continuous, but they are formed like a fragments. So these fragments are called as Okazaki fragments. So these Okazaki fragments are discontinuous fragments of DNA strands in the lagging strand. So what they have to do is they have to stitch them back. So the stitching work is done by DNA ligase. Okay, DNA ligase. Got it? So the DNA ligase comes and then it will fix the situation. So with the given options here, what happens here, guys? First one, we have is the helicase. Helicase comes first. After helicase comes, then comes the proteins. After proteins come, excuse me, I'll just erase this to make some space. Okay. After the proteins come, what do we have? We have the DNA polymerase, we have the primase. After the primase comes, we have the DNA polymerase. After the polymerase comes, we know that we have a situation with respect to our lagging strand, so that will be done by ligase. Ligase will stitch up the Okazaki fragments. So which option is very favorable to us? It is option B. We never come across phosphorylases. So phosphorylases is not in the equation, so we reject that, and our option is option B. So you can also go for the odd one out. The odd one out among the all options is option B, so you can choose that too. Okay. So let's go for the next question. And our next question is very interesting again. The pedigree chart clearly indicates the recessive disorder. Which of the following pictures indicate the recessive disorder? So this was a question from question number 113. See, whenever you have a recessive disorder, for a recessive disorder to be expressed, they have to be in what state? They have to be in a homozygous state. So let us say that it is an autosomal recessive disorder. So that means both autosomes are affected in the female as given in the picture, and then the male is fine. Let's say the male is fine. If the male is fine, then the next generation there is no way that the next generation would have this disease. Correct, right? So look at this picture. You can clearly make it out that if I translate this into next generation, what would I get? I will get A dash A, A dash A, XX, A dash A, A dash A, XY. Okay, then again A dash A, XX, A dash A, XY. So here we can see that both the XX and XY individuals are carriers of this disease. Now if they marry with each other, siblings marry, then what do we have here? Then the next generation, this is the first generation where none of them express the disease, they look like a carriers of the disease. But if you see what happens in the next generation, it's very simple. You have A dash A XX, sorry, A dash A XX, who is marrying with A dash A XY. Then you can see that there is a possibility that you have A dash XX and A dash XY, and then you have a normal A and A XX and a normal A and A XY. You can see here, okay, A dash A XX and A dash A XY. So here you can see that, sorry, this is right actually. Yeah, here you can see that the female would be affected. Okay, so that is why this particular disease is a representation of a recessive disorder. The option is option B for this particular question. Okay, so let's go for the next question. And our next question is, in DNA fingerprinting, question number 114, it says that, assertion says that the DNA fingerprinting is applied in paternity testing in case of disputes. Reason, it employs the principle of polymorphism in DNA sequences, as polymorphs are inherited from parent to the child. So again, you know the assertion and reasons. So these are the four options with respect to assertion and reasons. So let's look at the first statement. So can we use DNA fingerprinting for paternal issues? So what are paternal issues? Paternal issues are the cases where there is a doubt of who is the parent. Okay, whenever there is a doubt, when a mother claims that this is the father and he is not the father, something like that, when the court has to decide who is the real father, they go for DNA fingerprinting. So the DNA fingerprinting is employed to test the match, okay, how much of match is there with the parents. So that is very, very clean and that is very, very accurate. Now how do we do this? See, majority of the expressing genes in our human body, the expressing genes means whatever we express, like the skin pigments to the eye, to the site of the every single expressing genes are very, very similar to all the human beings on the planet. You cannot differentiate one human being with another human being with a functional gene or the genes which are acting or functioning. So what are we using here? We are using something which do not function at all. So these non-functioning genes are present in the mini-satellite and microsatellite regions as polymorphic sequences. See, majority of the chromosome is made up of non-functioning DNA or heterochromatin regions, but there are specific sequences in these regions, especially in the mini-satellite and the microsatellite regions, which we call it as tandem repeats. We call it as short tandem repeats, that is STRs, okay? STRs, okay, that is short tandem repeats, okay, single nucleotide tandem repeats, or we can call it as polymorphic sequences. So what are these? Why do we call it as polymorphic or repeats, tandem repeats? The reason is, if you see that if you have one generation and to the another generation, let's say it's me, so for me the number of repetitions of a sequence in the heterochromatic regions, for example, it can be AG, AG, AG like this, the repetition, okay, would be having a unique number. And these sequences are very unique to an individual, and it only matches to a certain percentage with the parents. Otherwise, it would not match at all with the random person. So what we do is, we call it as polymorphs because from one generation to another generation, these highly mutagenic regions, they do not appear with the same number, but they tend to match the regions of their parents. You understand my point? You can't get the exact number of repetitions, but you can get the sequence, like the patches of repetitions with respect to the father. So this match is around, accuracy is around 48 to 50% with respect to one parent. For example, mother 50% and father 50%, right? So around 45 above percentage would prove that that is the father. So we use polymorphic sequences in the DNA which are present in the mini-satellite and the microsatellite regions. So basically we refer them as variable number of tandem repeats or short tandem repeats. So these repetitive sequences are used to see the match of how much of these sequences are present with respect to father and the son. If the sequences are equivalent to above 45%, then he is the dad. If it is not, if it is below, then if it is not at all the father. Okay, that is how the code decides. So with respect to question number 114, both assertion and reason are true, and the reason is the correct explanation for assertion. So we go for option A. Okay, let's go for the next question. And our next question says, match the following column with respect to correct statement. We have column 1, column 2, column 3, and here are the given options. So let's look at them. Okay, so we have our column 1, that is mitochondrial matrix. So and then we have hemicellulose, chloroplast and nuclear lamella. Then we have thylakoid, mitochondria, plant cell and then nucleus. Here we have site of Krebs cycle reactions, polysaccharides organized in stacks of grana, and then we have primary site of DNA transcription. Let's look at it. Mitochondrial matrix is present where? It is present in mitochondria, so I'll represent it as A. So mitochondria is a site of what? It is a site of Krebs cycle, so I will represent it as Q. Got it? Next one. Hemicellulose, where is it present? It is present in the plant cell wall, so I'll represent it as B. What is plant cell hemicellulose? Hemicellulose is nothing but polysaccharide, so I'll represent it as R. Next one, we have chloroplast, and the chloroplast contains what? It contains thylakoids, so I'll represent it as C. And thylakoids are what? They are nothing but they are the organized stacks of grana, so I'll put it as P. Next we have nuclear lamella. Nuclear lamella contains what? It contains nucleus. Okay, then I'll put it as D. What is nucleus? Nucleus is a primary site for DNA transcription, so I'll write it as S. So what is our right option? The right option for question number 116 is option C. Okay, it's a very simple question, but the thing is you have to just look at the question very carefully. Got it? So let's go for the next question, guys. Our next question is, again, match the following column 1 with column 2 and select the correct options. We have the shapes of the chloroplast. In the other side of the column, we have the algae. So the shapes are given a cup shape, girdle shaped, okay, stellate shape, and then we have the reticulate shape. Here we have Eulothrix, Oedogonium, and then Chlamydomonas, okay, and Zynema are the examples. Okay, so what are the correct sequences? So if you look at the cup shaped are found in Chlamydomonas, so I'll write it as A. And then you have the girdle shape is found in Eulothrix, so I'll write it as B here. And then you have stellate shape, the stellate shape is found in Zynema, so I'll write it as C. And then you have reticulate shape. Reticulate shape of chloroplast is found in oogonium, so I'll write it as D. So your correct sequence for the match the following for this question should be option B. Okay, students, just take a note of it. This is a very important question with respect to shape of the chloroplast. I'll repeat again. You have the cup shape found in Chlamydomonas. Girdle shaped is found in Eulothrix. Stellate shape is found in Zynema. And then you have your reticulate shape, it is found in Oogonium. Okay, so let's go for the next question. Which of the following statements is false with respect to the viability of mammalian sperms? Question number 125. So the options are: sperm is viable for only 24 hours, up to 24 hours. Survival of sperms depend upon the pH of the medium and is more active in alkaline medium. Viability of sperm is determined by its motility. Sperms are typically suspended in a liquid medium. Okay, so these are our options. So let's look at each option. Which of the following statements is false with respect to sperm viability? Sperm is viable for only 24 hours. So this particular statement is wrong. The sperm can be viable up to 72 hours. Okay, so who is viable for only 24 hours? 24 hours viability is for ovum. Okay, ova or ovum. Ovum is the one that has only 24 hours of viability. So that is why this statement makes wrong. This particular statement is wrong. Since we have got already the answer, that is option A is wrong, the rest of the options are very, very correct. That means survival of the sperms depend on the pH of the medium. Exactly right. So usually the sperm's pH is alkaline. So that's why they're quite active in alkaline media. Then viability of sperms is determined by motility. That's also true. Sperms are typically suspended in liquid medium. That is the semen. You know that, right? So that is why our option D is also correct. Our next question. The first sign of growing fetus is noticed by. Here are our options. Option A says formation of digits in embryo. Option B says implantation of blastocyte within endometrium. Option C says listening to the heart sound carefully through the stethoscope. And then option D says the first movement of the fetus. So by this you can determine that you already have the first growing signs of a fetus. So let's see. So first one is the formation of a digits in the embryo. So the formation of a digits in the embryo basically takes at least 8 to 12 weeks. Okay, this happens between 8 to 12 weeks. Got it, guys? So this is the first thing. Next one, you have the implementation of blastocyte within the endometrium. Here you are not certain whether it has led to the complete implantation or the fetus is successfully growing. You need a proper establishment of placenta. Placenta should develop, okay, and there should be a proper integration between the embryo as well as the endometrial layers. Once this integration is done, then we can say that this can be a sign for the fetus to further grow. So this also cannot, this is too early. I can say this is very, very early to say that whether the fetus is developing or not. Next one is listening to the heart sound carefully through the stethoscope. Definitely yes, you can do it after 4 weeks. Okay, after 4 weeks to 6 weeks, you can clearly see the murmur. Okay, so that is possible. First movement of the fetus. So this is a very later on phases. This happens, let's say that it's between the 24 weeks to 30 weeks easily. So this would take 24 weeks to 30 weeks. That means almost 4th to 5th months of pregnancy should be achieved to feel the first movements of the fetus. So that's why our correct option is you have to listen to the heartbeat of the baby through the stethoscope. Then it is possible to determine the developing fetus. Okay. Next one. We have 132 question number 132. Here we have an assertion. Primary spermatocytes of testis are haploid. Next one, we have the reason. Primary spermatocytes are formed by meiosis 1 in the spermatogonia. So if both statements, if both assertion, same thing, assertion, reason are true, reason is the correct explanation for assertion. Then again, if both assertion and reason are true, but reason is not the correct explanation for assertion. If assertion is true, but the reason is false, if both assertion and reason are false. So what happens here? During spermatogenesis, we have spermatogonium. The spermatogonium first goes under mitotic division to, the mitotic division means you have the 2n, right? So the 2n becomes more and more, and they make more spermatogonia. Some of the spermatogonia, they do not complete, actually if you see the proper process, they do not complete a proper meiosis 1. They suspend at a certain stage, which we call it as primary spermatogonium. So the primary spermatogonium are not haploid, they are diploid. And next one, the primary spermatocytes are not formed by meiosis 1. They are a resultant of mitosis modified cells. Some of the spermatogonia modify themselves into primary spermatocytes. So this is also wrong statement. So our option is if both assertion and reason are false. So your correct option should be option D. Okay. So let's go for the next question. You have 133. Match the following. So in column 1 and column 2, let's see, in column 1 they have given us hypodermis in dicot stem, pericycle in dicot stem, ground tissue in monocot stem, and then phloem parenchyma in monocot stem. Then you have absent, then you have parenchymatous cells, collenchymatous cells, and then you have sclerenchymatous cells. So let's see, what is our right option here? First one, hypodermis in dicot stem. When you see the hypodermis in dicot stem, a hypodermis is made up of collenchymatous tissue, so I'll write it as A. Next one, you have the pericycle in dicot stem. The pericycle in the dicot stem is made up of sclerenchymatous tissue, so I'll write it as B. Next one is the ground tissue. The ground tissue in the monocot stem is made up of parenchyma, so I'll write it as C. Then phloem parenchyma in monocots is absent, so we go for D here. Okay. So if you match the correct sequences, the correct answer should be C. So I'll write it as C. Okay. 133. The right option is option C, guys. So it's an interesting picture, but a very easy one. So let's identify the phases from the graph given below that shows the change in the DNA content during various phases of the mitotic cell division. So we know that a cell division can be divided into several phases. Usually the cell cycle can be divided into several phases, and in this phase, okay, so basically it starts from G1, and the G1 is a short phase. Again you have is the S phase, the synthesis of the DNA phase, and then you have is the G2 phase, and then you have an M phase, then you have after M phase you have a G0 phase. Okay, G0 phase. And here the list is given, and we have to find out with respect to the DNA volume, that is 2C to 4C, etc. So what is happening here, guys? Question number 135, the right option would be option C. You can see that A is represented as the G1 phase. Yes, B can be given as synthesis phase. You can see here by the end of synthesis phase, the volume of the DNA is doubled, that is 4C. And then you have is the C. C would be the G2 phase, and then the D would be the M phase. Okay, the M phase. Clear? So this is about your question number 135. The right option is C. Okay, students. So let's go for the next question. Which of the following statements is incorrect? Parthenium or carrot grass causes pollen allergy. Vegetative cells of the pollen has abundant food reserve. All pollen causes severe allergies and bronchial affections. Then sporopollenin is the most resistant organic matter known. And our options are, option A says all of these, option B says only B, option C says only A, option D says only C. So let's look at it, each statement one at a time. We have to find the incorrect statement. Parthenium or carrot grass can cause pollen allergy. Yeah, the biggest culprit for the pollen allergy is this particular plant, where the parthenium releases a lot of wind pollen, and that can cause a severe allergic reactions. Okay. Next one, vegetative cells of pollen has abundant food reserve. So vegetative cells of the pollen grains do have the abundant food reserve. Why? They have to support the development of the pollen tube, as well as, okay, they have to make sure that it reaches, okay, the male gametes reaches the ova. That's why they have the abundant food reserve. Next one, all pollens cause severe allergies and bronchial affections. This is wrong. Not all pollens causes pollen allergy. Only some pollens cause pollen allergy. So this particular statement is wrong. Next we have the D, that is sporopollenin is the most resistant organic matter known. That is also true. So the option is option D, that is only C. Okay, students. So let's go for the next question. Juxtaglomerular apparatus is formed by the cellular modifications in question number 141, and our options are afferent arteriole and DCT, that is distal convoluted tubule. Option B says efferent arteriole and proximal convoluted tubule. Option C says afferent arteriole and proximal convoluted tubule, and option D says efferent arteriole and distal convoluted tubule. Got it? So what is JGA or juxtaglomerular apparatus? This is like a sensor. So what happens, you know that this is our glomerulus, correct? No, in the nephrons we are talking about. So one of the arteries enter the glomerulus and one of the arteries exits the glomerulus. Why? The blood from the body enters the kidneys, and as they enter the kidneys, they divide and divide and divide. They become arteries to arterioles, arterioles to small blood capillaries. So these small blood capillaries eventually enter into this bowl called as Bowman's capsule, where they divide and where they become very highly coiled structures. Okay, so together we call it as a glomerular capsule, correct? No, here what is happening, the one that brings the blood is called as afferent. Afferent is the one who is bringing the blood from the body to the kidney, and then efferent is the one that takes the blood out of the kidney. So here what happens is, there is a last part. See what happens here, afferent comes, efferent goes out. Here between that there is a very coiled structure, okay, and finally this coil exits in the form of what? PCT, that is proximal convoluted tubule, then loop of Henle, LOH, that is descending loop, ascending loop, and finally it becomes what? It becomes DCT. What is that? Distal convoluted tubule. So the distal convoluted tubule is the latest or the last portions of the nephron. Here it is determined what to save, what not to save. Whenever you have the very less water content in your body, then the urine formation has to be reduced. Whenever you have very high water content in your body, then the urine formation can be high or it can be moderate. So to monitor this, there is a special type of cells that are formed between the junction of your DCT and the afferent arteriole. Now what happens, this is your afferent arteriole, okay, and this is the DCT. Between DCT and the afferent arteriole, there are special type of cells that colonize here. What they do is, they project the cilia inside it and then they monitor the blood, okay, the blood flow. Here they can recognize what to be saved, what not to be saved. In case the blood pressure drops, they insist on increasing the blood pressure. In case the blood pressure increases, they insist on decreasing the blood pressure. So this monitoring action is done by something called as juxtaglomerular apparatus, and the pathway or the mechanism they undertake is called as RAS pathway. RAS is also called as renin, aldosterone and angiotensin renin, aldosterone and angiotensin pathway. We'll discuss about this pathway in very detail, but in simple terms, the role of JGA is to balance the BP, blood pressure or the blood flow into our nephrons. Okay, simple. So that is done between the afferent arteriole and the distal convoluted tubule. So our option is option A. Do not forget this concept. Many times this question has appeared in our past papers. Okay, next one. How many of these given items given below in the box are viral STDs? Okay, that is viral sexually transmitted diseases. We have hepatitis B, AIDS, gonorrhea, genital herpes, syphilis, genital warts and chlamydiasis. So here the option is 4. Okay, the option is there are 4. I can see that there are 4 diseases which are virulent diseases. But I want to give you very important information in the following slide. Please pause the video and observe this slide so you get a complete information on STDs, which are curable, which is non-curable, which is bacterial, which is viral, etc. Let's see here. Gonorrhea is a bacterial disease called as Neisseria gonorrhea, which is curable. See, the first half is curable, the second half is non-curable. Look here. And then the following symptoms are given. You can pause and copy down the symptoms, okay? Next one. Syphilis is caused by a bacterium called as Treponema palladium. Then you have chlamydiasis, which is caused by a bacterium called as chlamydia. Then you have genital warts, which is caused by a virus called as human papilloma viruses. Okay, so HPV, another name is also called as HPV virus. Next one you have is trichomonosis. Okay, so trichomonosis is a protozoan disease, very important, please mark it down. So it causes a disease called as trichomonosis is a disease. It is caused by a protozoan called as Trichomonas vaginalis is the causative agent, which is a protozoan. So all this one, which one? Gonorrhea, syphilis, chlamydiasis, genital warts, as well as trichomonosis, when they are diagnosed at a very early stage, they can be cured very successfully. Okay. Next one, you have incurable diseases. Here one more thing, just please observe that genital warts is the only viral disease that is given here. Okay. Second list, you have HIV. Okay, AIDS, that is acquired immunodeficiency syndrome, which is caused by a disease called which is caused by a virus called as HIV virus. What is that? Human immunodeficiency virus. So this is also a viral disease. Okay. Next one, you have genital herpes. Herpes simplex virus is a causative agent. So this is also a viral disease. Next one, you have hepatitis B. Hepatitis B virus, this is also a virus disease. So 1, 2, 3, 4. 4 are viral diseases, and the respective bacterial or protozoan diseases is given, and their following symptoms are given. You can pause this video and I can just check on it. It's a very important slide. It will shortcut your work on studying the whole lot of syllabus in your textbook. Got it? So let's go for the next question. 146 question. Match the following column 1 with column 2. So we have column 1 with the cell phases, cell division phases with respect to mitosis, and then you have the column 2. So let's look at it, guys. First one is the prophase, metaphase, anaphase, telophase, and cytokinesis. So what is prophase? According to the given question, you know that the prophase is the first phase where the chromosomal material condenses to form a compact mitotic chromosomes. So that is why I put it as A. Now what is metaphase? Metaphase means what happens? The spindle formation is successfully done and the spindle fibers go and attach to the kinetochores. So I'll write it as B. Next one is your anaphase. Anaphase, what happens? Usually these attached chromosomes, they start moving apart from each other, where your centromeres start splitting and the chromatids start separating. So I'll write it as C. In the telophase is the last phase where you have seen all the chromosome material has migrated to the opposite poles and the spindle fibers disappear, and eventually the nuclear envelope starts reappearing. So that is what is your telophase. Sorry, I should have written as PQRST. So I'll write it here. Okay. So this is P, this is Q, and this is R. And finally, you have cytokinesis. In cytokinesis, what do we have? We have the formation of the cell plate. Okay. For the question number 146, the right option would be option D. Okay. The right option is option D. Please go through the cell cycle and cell division chapter. Each phase, each topic is very, very, very crucial. A large amount of questions come from this chapter. Okay. Next one. Read the following statements with regard to frog and choose the correct option. Statement 1 says frogs are cold-blooded and uricotelic animals. And then statement 2 says in male frogs, the ureters act as a urinogenital tract. So we have to choose which statement is correct, which statement is wrong. Got it? So let's look at the first statement. In 149 question, the first statement says frogs are cold-blooded. Correct? Okay, they are poikilotherms and they are uricotelic. Are they uricotelic? No. Why? Because they secrete their metabolic waste as urea. So they are ureotelic, not uricotelic, they are ureotelic. That is why our statement 1 is wrong. Second one, statement 2, in male frogs, the ureters acts as a urinogenital duct. Yes, correct. In male frogs, the common duct is used as both for urine as well as a genital duct. That's why the statement 2 is correct. So what do we have here? Statement 1 is incorrect, but statement 2 is correct. So our option B is correct for the question number 149. Okay. So let's go for the next question, guys. Select the 2 correct statements out of 4 statements given below with respect to lacoperon. So lacoperon, that is gene expression with respect to prokaryotes. We would have studied this. So let's look at the statements. Glucose or galactose may bind with the repressor and inactivate it. In the absence of lactose, the repressor binds with the operator region. The LAC-Z genes codes for permease. This was elucidated by Francis Jacob and Jacquin's monad. So we have to choose the correct statements. Okay. See here, guys, what happens here when we look at the lac-operon model was elucidated by Jacob and monad. Okay, so this is absolutely correct. What they did was they wanted to see how the gene functions or who controls the activities of the gene. So during this time they understood that the gene of a prokaryote is present as a monocistronic gene. That means one promoter controls several genes. Okay, so we have a promoter region, and then between the promoter region we have an operator region, and then we have a structural gene. We have LAC-Z, LAC-Y and LAC-A, like this. So the LAC-Z, this promoter is where the whole transcription starts, and then the operator is where the regulation part will happen. Usually this operator is blocked by a sequence called as repressor sequence. Okay, this is a repressor sequence. And then you have the three genes which are sequentially arranged. The LAC-Z is responsible for the production of beta-gal. LAC-Y is responsible for permease. And the LAC-A is responsible for the production of trans-acetylase. Okay, trans-acetylase. Now what is happening here, guys? See, this particular gene do not function whenever there is other source of food. The usual source of food for the prokaryotic cell is glucose or galactose. Whenever they are there, this LAC gene is the one that is helping this bacteria to break down lactose. So when they have easy source as glucose, then why would they go and break down lactose? No, right? They need to be switched off. To switch off what they have a repressor element. So this repressor element usually goes and blocks the operator region. How does it block? Whenever there is no allolactose. You see here, allolactose. So whenever this allolactose do not bind to the repressor, then the operator is always blocked. Understand this. When allolactose do not go and attach to the repressor element, then the operator is free. Whenever the operator is blocked actually. You get my point? So you see here, glucose and galactose may bind with the repressor and inactivate it. That is not true. Only lactose or allolactose. So they can only go and bind to the repressor. Why? Because the repressor is very sensitive to the lactose or allolactose. The allolactose goes and binds to the repressor. Now once they form these structural modifications, now the repressor cannot recognize the operator region. When they cannot recognize the operator region, what happens here? The operator becomes free. When the operator becomes free, what will happen here, guys? Now the gene expression will start. For the gene expression, that means whenever they can activate or inactivate, who is doing? The allolactose is doing. The glucose and galactose do not have a role. Got it? Next one, the absence of lactose, the repressor binds with the operator region. This is true. Whenever there is no lactose, now what will happen? The rest of the lactose is totally consumed. Even this is the repressor. Usually the allolactose is bound to the repressor. When there is no lactose, this allolactose releases the repressor. When the repressor is released, what does this repressor do? Repressor will go and block the operator region. When the repressor will go and block the operator region, what will happen? Now they will, in the absence of lactose, the repressor will bind to what? Operator region and stop the function of the LAC gene. Next one, the LAC gene the LAC-Z gene codes for permease. I told you the LAC-Z gene is responsible for the production of beta-galactosidase, whereas LAC-Y is responsible for the production of a protein called as permease, whereas LAC-A is responsible for production of trans-acetylase. Got it, no? And fourth one, I already told it is true. So which of the statements are true? 2 and 4 are correct. Where do we have 2 and 4? Option C. So for this question, option C is correct. Next one, match the following and choose the correct option. You have group A and you have group B. Let's see. In group B, we have aileron layer. What is aileron layer? Aileron layer is usually seen in monocots and they are found as a covering of the endosperm. So I'll write it as A. Next, you have parthenocarpic fruit. Any fruit that is found without fertilization is called as parthenocarpic fruit. I'll write it as B. Ovule. Ovule is a result of the seed formed. Okay, seed. Ovule develops into seed, simple terms. Okay, and then ovary develops into fruit. Correct? No. So I'll write it as C. Endosperm is formed by the double fertilization. So what is our option? 151. In 151 our right option should be B. B is the correct sequence. You just go through this. Okay. Next one. So here is a pedigree chart that shows the inheritance of which of the following Mendelian disorders. So here there are two parents. Okay. So next what will happen? You can see that in the next generation you have only the son and daughter are expressing this and the rest of the sons and daughters are fine. So how is it even possible? So let's assume that we have a case where we have autosomal recessive disorder and XX will marry A-A-A-A, XY. Where can we find this condition? Whenever there is an autosomal recessive trait, this condition can be seen. So if you work out the pedigree chart, what do we get, students? We get A-A-X, X. Then we have A-A-XY. Then we have A-A-XX. Sorry, A-A-XX and A-A-XY. Okay. Then you can see here A-A-XA, okay, A-A-XX, A-A-XY. So you can see here, according to the pedigree chart, who will be expressing? Whenever you have an autosomal recessive trait, both of them should be in the homozygous situation. So you have a male and you have a female here. Sorry, it should be Y. So you have a male and you have a female. See, this male and this female is expressing. And among them, these two are the carriers. Among these two are the normal ones. So which of this disease? Autosomal recessive disease. Which is the example given for autosomal recessive disease here? Sickle cell anemia. So our option is C. It's very easy to work out the pedigree pictures. So you need a little bit of practice. We'll do it more and more. Okay. Next one. Among the following items listed below, those that are not technically correct names of the floral ole are what? Very, very simple question. So here you see the list is given of the floral ole: androecium, carpel, corolla, sepal. So we have to choose. Androecium is for male gamete. Corolla is also called as gynoecium. And then corolla is correct. Okay, calyx. So what are the four parts? Androecium, gynoecium, calyx, corolla or calyx. So which of them are option? We don't call it in scientific terms as sepal, and here carpel is also a common term. So our right option, our wrong options are 2 and 4. We should see which of them are not technically correct. So the 2 and 4 are not correct. So the 2 and 4 is option C. So question number 160 is option C. So very simple one. When you look at the flower pictures, you will see sepals and petals. So usually you will forget it. So androecium, gynoecium, calyx and corolla are the 4 parts. Okay. Let's see the next one. 4 different steps that occur during the mitosis are given below. So we have to choose which are the proper sequence. The first one is cells do not show Golgi complexes, endoplasmic reticulum, nucleolus and the nuclear envelope. Nucleolus, Golgi apparatus and ER, endoplasmic reticulum, reform. Then we have chromatids move to the opposite poles. Chromosomes are moved to spindle equator and they get aligned in the metaphase plate through the spindle fibers in both plates. So what happens first? When you see the prophase, the first phase of the cell division or the nuclear division, what is happening here? You can see the disappearance of the nuclear membrane and the dissolution of nucleolus. Along with that, very important organelles also disappear. That includes Golgi complexes, endoplasmic reticulum, nucleolus and nuclear envelope. All of them disappear in the first phase. So this is the first event that happens. After this first event, what happens? Usually the chromosomes are the ones that condensed chromosomes, they get attached by the kinetochore and then they align at the metacenter. Okay, they align at the metacenter. So that is, chromosomes are moved to the spindle equator and they get aligned at the metaphase plate through spindle fibers to both poles. So once they attach to the spindle fibers, now the migration starts. So that should be the end of metaphase. Now what happens? The third, this is the second phase. The third phase should be chromatids move. They should be moving to the opposite poles by anaphase. By telophase, what will happen? The already moved chromosomes, they start developing nuclear envelope, and chromatids, sorry, they start developing a nuclear envelope along with the reappearance of the cell organelles like nucleolus, Golgi complex and ER forms. So this is the fourth one. So what is our following sequence, guys? 1 4 3 2, 1 4 3 and 2. So option C is correct. Okay, option C is correct for 161. Chalo, let's go for the next question. Okay, during water stress, the bulliform cells become what? They become turgid, they become flaccid, make the leaves curl inward, make the leaves exposed to the surface. So our options are both A and C, B and D, A and D, and B and C. What are bulliform cells? Bulliform cells are dumbbell-shaped cells. Usually they are found in monocots. Okay, so monocots are the examples of grass. You might have seen that some of the grass species, whenever there is absence of water during the summer, the leaves are not like flat, they are curled like a tube. So why do that happens? Usually these bulliform cells are the ones that change the morphology of this leaves exposure based on the availability of water. Grass species are very well adapted to dry areas. So what they do is, to prevent the transpiration loss, like the loss of water, these bulliform cells, they lose the water that is present in them, so that the leaf loses the rigidity, and instead of becoming the flat, they start curling down into a small tube. Okay, so that is when you see that whenever there is water stress, okay, that is lack of water, the bulliform cells become flaccid and then they curl inward. So our option is B and C, that is D. Okay, the right option for the question number 166 would be option D. So let's go for the next question, that is question number 169. Select the incorrect statements among the following. Ring arrangement of vascular bundle is a characteristic of dicot stem. Phloem parenchyma is absent in monocots. Monocots have sclerenchymatous hypodermis. In monocots, peripheral vascular bundles, which are generally large, okay, and then you have centrally located small and water containing cavities are present within the vascular bundles. So let's see which of the following is incorrect statement among this. First one, ring arrangement of vascular bundles is a characteristic feature of dicot stem. This is absolutely correct. So it is arranged in the form of rings. Next one, phloem parenchyma is absent in monocots. That is also correct. The phloem of the monocots usually have only three parts of phloem. Among them, phloem parenchyma is absent. Anyways, next one, monocots have sclerenchymatous hypodermis. This is also true. Next one, this is very, very important. You can see here, usually the peripheral vascular bundles, which are generally large, and then you have centrally located vascular bundles, which are small. So you have the water containing cavities are present within the vascular bundle. See, water containing cavities are not always present. They are usually present in maize species. Okay, in maize. Okay, so usually they form this. The cells will dissolve and they form into a water cavities. It's not the proper or characteristic feature of all monocot. Monocots. Only in some monocots like maize you can see here. And the next one is the arrangement of this vascular bundles is quite scattered in the beginning, and then usually you have the arrangement of peripheral bundles which are large and then centrally located which are small. So our option D is wrong with respect to 169th question. Okay, let's go for the next question. Okay, if haploid chromosome number is 10, the monosomic number shall be 9, 18, 11 and 19. So let's look at which is the right option. See, haploid chromosome number is 10. So the formula for the haploid chromosome is 2n minus 1. So this is the formula to find the monosomic condition. So that here in haploid state, n is the number of haploid chromosome. Okay, haploid chromosome. Okay, so what do we have here? 2 into 10 minus 1. So what do we have? 19. So what is monosomy? Monosomy is a condition where there is a loss of one chromosome in a diploid state. So the option should be 19. So what is monosomic? Monosomy is if I have 46 chromosome in my diploid state and eventually I get only 45. If my only 45 are there instead of 46, then I am monosomic. So there is a loss of one chromosome. Got it, right? That is monosomy. So 2n minus 1 is a formula. Next one. In plant, red fruit R is dominant over yellow fruit and tallness is dominant over the shortness. If plant RR capital T small t genotype is crossed with the plant small r, small t, what would be its next generation? So let's look here. Okay, we have capital R, capital T, small t, guys. Next one, we have small r, small t is another one. Correct, right? So what do we have? Capital R, capital T is one type of gamete. Capital R, small t is another type of gamete. Next one, small r, small t is another type of gamete. So when they reproduce, what do we have? Capital R, capital T, small t, capital R, small r, okay, sorry, capital R, small r, then we have small t. Okay, so this is the possibilities: capital R, capital T, small t, capital R, capital T, small t. So these are the possibilities in which we can get. So the phenotype is 50%. Okay, 50% will be tall and with red fruit. Okay, so you can see here. Here what do we have? We have 50% are tall with red. Again, we have here capital R is tall, no? The capital R and small r would be capital R would be is red flower. Okay, this is red and short. Sorry, this is red and short. So what is the ratio here, guys? 1:1. 1:1. So what is the answer? 50% will be tall with red fruit. Okay, 50% will be tall with 50% will be short with red fruit. Okay, so that is the answer. So let's go for the next question. Although much is carried in blood, yet blood does not become acidic because. It's a very simple question, but a small additional information is required. It is absorbed by the leukocytes, and then blood buffers play an important role in the transport. It combines with water to form, which is neutralized. Okay, which is neutralized by. Then it is continuously diffused through the tissues and it is not allowed to accumulate. So if you look here, the blood pH balance, okay, is highly maintained by your buffers. What are buffers? Buffers are any chemicals that alter the pH by a very minute range. Okay, so we have blood buffers like sodium bicarbonates, okay, or potassium bicarbonates or any of the ions. So what they do is they can act like a buffer. Either they increase the acidity or increase the alkalinity based on the situational demands. So blood buffers play a very important role in managing, okay, the pH balance of our blood, okay, and not letting it. So that is option B, 187. Next one. 195 question. Even after killing a generative cell with a laser beam, the pollen grain of the flowering plant germinates and produces normal pollen tube because. Option A says laser beam stimulates pollen germination and pollen tube growth. Laser beam does not damage the region from which the pollen tube emerges. The contents of the killed generative cell permit germination and pollen growth. The vegetative cell has not been damaged, that's why it still germinates. This is a very simple question, but I have seen this question several times in the Neet papers. Okay, look here. If I kill the generative cell, you know that the pollen grain is made up of two cells. One is the large vegetative cell and a small generative cell. The small generative cells is responsible for the formation of male gamete, okay, whereas the vegetative cell is responsible for the formation of tube cells. Okay, it is responsible for the formation of tube cells. You get my point, guys. Now, if I destroy this generative cell, the vegetative cell is still function. So what it does, it will start producing a healthy pollen tube. So our option is the vegetative cell has not been damaged. So it will be producing a pollen tube. Okay, got it? So all right. So we come to the end of this question paper discussion. Thank you very much, students. I hope to see you in the next video. Until then, please watch this video. If you have any doubts, please get back to us. Have a nice day.